How is everybody? All right. I apologize for how close the air feels in this room, but that's what we got. All right. So sometime today, I think the due date on the assignment is midnight or something tonight, but please don't stay up that late. I just want you to submit through the portal on the Moodle what your group is, who you're going to be working with, and your topics for your superfoods. Maybe like a sentence or two, kind of describing maybe the angle you're going for, or your overall idea. Don't just say, blueberries, and then move on. Although that would be a great start. Yeah, blueberries are wonderful. They're here in Maine, and they're certainly thought of as a superfood. But tell me, what about blueberries? Just give me like a sentence or two about it that helps me know that your mind is heading in the right direction with that. And is there anybody who is planning on recording at the Common Grounds Fair? Anything? Is there anyone who's got a topic related to that? All right.
Possibly, if we stumble upon something that fits.
What's that?
I don't know, if there are take a recorder just in case.
Yeah, that's what I'm saying. I'm encouraging people to grab a recorder if you think there is any chance that, there's going to be a lot of different superfoods that are actually grown in Maine, right? There's people that grow kale. There's people that grow elderberries and blueberries and garlic, and a lot of foods that have at one time or another been labeled as superfoods. So I don't know if those are any of you have chosen those topics, or just superfoods in Maine in general, or the Common Ground Fair as a vehicle for superfoods. I don't know any of that. Yeah.
How are we getting to the Common Ground?
Okay. So first, the thing about the recorder. So if you want to bring a recorder, get one the way Tim told you. On Friday, I would like everyone to come to the lab, get a data point off their, tend to their sauerkraut and get a data point of pH. It should take you about 10 minutes, I'm hoping. And then the Colby certified drivers, raise your hands again. Okay. Well, I'm going to need 2 of you on Friday, and they don't have to be the 2 that drive on Saturday or even going on Saturday, but I'm going to need 2 of you on Friday to help me pick up the SUVs. So there are going to be 2 SUVs that I want to get during lab period. So I don't know who that is, but does anyone want to volunteer for that now? On Friday, you will. Okay. Oh yeah, absolutely. We will be back. I imagine we'll be back before 2. Probably like 1:45. Shouldn't take us more than 10 minutes to drive up there, grab the SUVs, and stick them in Bob's. So it should be a pretty quick trip. I just need some help doing it. Okay. So you'll take your pH points, and then I'll give you $5 and your ticket to the Common Ground Fair, because particularly for the people, maybe I'll just give that to the people who are not coming with us on Saturday. Who's going to be there on Saturday? All of you will meet at 9 o'clock. Does that seem like a reasonable time? In front of, let's meet in front of Olin there, between Olin and Bixler. And the 2 drivers, whoever is going to drive, who wants to volunteer for that on Saturday? So the people who are going on Saturday, who's a certified driver? Okay, well you're one. I need one more. Is anyone else going on Saturday that's a certified driver? Really, no one else. All the other certified drivers aren't going on Saturday. That's a problem. I have to figure that one out.
What do you mean by certified?
It's super easy. Do you have a driver's license? Yeah. You go over to PPD, you sign a form, you flash them your driver's license, and you sign some form, and that's it. It's just so that your name can get on their insurance waiver or whatever. So you just have to flash a valid driver's license. So are either of you guys going? Yeah, great. Yeah, go over to PP or the police department, I guess, over in what is that, Bob's? Yeah, and just sign up, and then you can drive. In fact, if you would do that, it would be great to help me and Zane on Friday. So we'll meet at 9 o'clock and take the vehicles. Everyone who's going, we'll load up in the vehicles. We'll drive out to Common Ground Fair. We're going to probably be stuck in the car for about an hour because it's only a half hour away, but these massive Saturday's the busy day. So we'll be stuck in traffic trying to park. And then we'll walk around for the day. I don't want to be there too late. I'd like to bolt before the crowd gets back on the road, so maybe like 2 or 3 in the afternoon, something like that. I don't know. We can figure it out when we get there, how long we all want to be there and how much time it takes. We should share cell phone numbers. We should get a class list of cell phone numbers of people who are going so that we can coordinate while we're there to make sure we all get out together. So that's that. There's a data sheet. There's like some questions that you'll see in the lab if you read the lab for this week. Your goal while you are there, besides collecting any kind of audio for the podcast, the primary goal is you're going to buy some apples, and you need at least 3 apples, preferably 6 apples. You have to look at the lab. I forget which I decided to do. I think you may need 6 apples of 2 different types, 2 different. Yeah. It says in the lab. I might be wrong. Maybe I dialed it back just to 1 variety, but I think it's 2 varieties of 3 apples apiece. You can buy more than 3 apples if you want, but I'm only giving you $5, which should be enough to buy 6 apples. Yeah, and if you can't, there should be lots of apples there. And if you can, get weird varieties so that we have a diversity in the class. Try to buy ones, like if everybody brings back Max, that'll be interesting because we'll have collected a lot of data about Max. But if you all go to the same farmer and get Max, then everybody's data will be exactly the same. So explore a little bit. And there are questions that you'll ask the farmer there, like you have to look through the lab, but it's, how long have they been farming? Is it an heirloom variety? Is the apple known by other names? Stuff like that. You'll see there's like a half dozen questions that you'll ask the farmer that grew those apples. And then we'll come back on Saturday after this, and we'll take the apples and stick them in the fridge up in the lab, and then you'll be done. So then the following week, we'll use those apples. We're going to cut them up, do a bunch of experiments with them, measurements on them. It's really a lab about measurement. And then I'll have brought my dehydrator in, and we'll dehydrate them and do some more measurements on the dehydrated apples. And then you'll have dehydrated apples that you got at the fair. Okay? So that's the next 2 weeks in lab. There'll be a lot bigger data sheet to fill out the following week in lab once we are actually in the lab with the apples. All right. And then I'm going to make a couple minutes at the end of class today to have you guys answer a few questions about the class, but I'll stop us when we're there. Are there any other questions? All right. Well, last time we were trying to walk our way through the periodic table and just try to understand the history behind the elements that make up the human body. We started the lab by having you guys try to guess what elements made up the human body. I did the same activity with my human anatomy class at the high school, and I'll tell you guys that they were nipping at your heels, but you did just edge them out a little bit in terms of being able to identify all the common elements found in the human body. So that was good. All right, and we left off with Joseph Priestley and oxygen. Joseph Priestley, the Unitarian minister that gave us bubbled water and nitrous oxide, also discovered oxygen until he was chased out of England at the end of his life for being a little bit too radical. He was kind of a radical. I guess it was all the nitrous oxide going to his head. And he fled to the United States and is buried in Pennsylvania, actually. You can see his grave there. So next on the list is sulfur. So sulfur is a profoundly ancient element in terms of the canon of chemistry. It's been known since the prehistoric times, which is why I've got a caveman up there saying he was a scientist. And sulfur had been first proposed as an element by Lavoisier in 1777 when he was a young man. We've talked about Lavoisier a little bit already. And when I say an element, we mean not a compound. And I'll talk about Lavoisier and his attempts to show what an element is and not a compound a bit more when we get to carbon. The word itself comes from the Latin sulfurium, which is a derivation of a Sanskrit word, solvere, and meaning the enemy of copper. And so that is an allusion to the fact that sulfur, copper sulfate, is sulfur readily forms a salt with copper, and something known by the alchemists for a long time. Sulfur is essential to the human diet, and that is because it is a component of 2 of the 20 amino acids that make up the proteins in our body. You see here both cysteine and methionine have sulfur in them, and this is important because it adds a structural element to protein called the disulfide bond. We'll talk about proteins and protein structure as we continue to add more and more science to the class here. But you'll see that the disulfide bond is one of the tools in the chemical bonding toolbox that biology uses to give proteins their 3-dimensional structure. So the Sicilians in Italy were extracting sulfur, recognizing its utility for centuries now. And here's a depiction of a kiln that they used to extract sulfur from the volcanoes in Italy. The symbol that you see on the right down there, there's a bunch of alchemal symbols that the alchemists used throughout the Middle Ages to denote sulfur. But the one down there is the symbol for brimstone. Brimstone, of course, is referenced in the Bible and is a corruption of the word Brennstein by the Germans. And it means stone that burns. So sulfur is an important component to gunpowder, and its chemical value, had been recognized for a long time. So we're going to put the caveman on the chart right there for sulfur. Sulfur. Disrupted first grade, popping stink bombs, starting fires, still can't spell your name. That's an allusion to the reference to the fact that sulfur can be spelled with an F or a PH. Both are correct. All right. So I've been going through all these different elements. This is where I actually wanted to start today. And I've told you they're important. They make up the human body, but how more specifically? And that's what we're going to investigate. So to begin talking about the crucial importance of elements, we're going to start with the food cycle. And this, this is probably something from 5th grade biology class that you guys hopefully remember from long ago. All life on Earth is enabled, is facilitated, the energy to make it happen comes from the sun, and the sun's radiation is absorbed by primary producers, plants, through a process called they're autotrophs. Autotrophy. Yeah, autotrophy is how you say that, pardon me. So this means that they're able to fix their own carbon. They don't have to find their carbon in some other source. They're able to take carbon dioxide, that's a gas in the atmosphere, and using the sun's energy, they're able to form sugars out of it. And then those sugars become the structural components and the energy. That's how they store the energy that they will use for the various cellular mechanisms, cellular processes that are part of their life cycle. The rest of us, everything else in the bottom there in the green box, that's everything else, are called heterotrophs. And heterotrophs are organisms that get their carbon elsewhere. They get it from the autotrophs. So primary producer is consumed by a primary consumer, such as an herbivore. You've got squirrels, cows, grasshoppers, all kinds of different primary consumers. You move up the food web to secondary consumers. These can be omnivores or carnivores. So an omnivore is some kind of creature that eats both plant and other animals. Carnivores only eating animals. And then you get to apex consumers. And they can also be omnivores or carnivores. And these are animals that are essentially predators that eat anything below them on the food chain. However, it is a cycle, and entropy rules the universe, meaning that all good things come to an end, everybody dies, and then decays and becomes the province of what are called the detritivores. These are the decomposers in our ecosystem. They include fungi, different kinds of molds and mushrooms. It includes insects, detritivore insects like flies that eat rotting food, rotting organic material. Bacteria and annelids, worms, are all part of the decomposers. They break down the organic carbon and provide trace nutrients that can be taken up through the soil by primary producers, such as that plant in the picture right there. So that's the food cycle. And we're going to look at how the different elements, some of the different elements lay on top of this food cycle and how they themselves cycle through the ecosystem. Because there are, we haven't talked about some of the big players yet. We haven't talked about carbon. We have not talked about nitrogen. We haven't talked about phosphorus. We're going to talk about all those. Those are some of the most important elements to life. And we're going to see how those cycles lay on top of the primary food cycle. All right. So the three most important elements I just mentioned are carbon, nitrogen, and phosphorus. So this is the story of the Redfield Ratio. Here's this guy named Alfred Redfield that lived around, he was most prolific during the interwar period, but between World War I and II. He was interested in the oceans primarily. And what he did, his first observation was to look at different concentrations of various elements in ocean water and compare that to the elemental ratios in the algae that was floating in the ocean. And surprisingly or unsurprisingly, I'm not sure, those ratios were remarkably constant throughout the oceans of the world. He traveled all over the world in this boat called the Atlantis. And this guy was from Cape Cod, a dowdy New Englander there. And he assembled a team who actually were not particularly seaworthy. Apparently, there are stories of him going out on this boat called the Atlantis and taking water samples from the Atlantic and the Pacific and different oceans around the world, particularly in the Indian Ocean, which is one of the roughest oceans in the world. The waters were so choppy that they had initially the team of scientists had a really hard time on the boat because they were all seasick and weren't able to get as much work done as they wanted to. But eventually they collected the data and they came up with this carbon to nitrogen to phosphorus ratio of 106 to 16 to 1. That number is held up remarkably over time. I think the current Redfield ratio that science gives us it's like 117, or maybe it's 114 to 17 to 1. I think that's the actual Redfield ratio that people cite now. So what is this? What is this telling us? And what forms are these elements taking as they get into the plant? So thinking about it terrestrially, here's our plant. It's going to be absorbing its carbon, that 106, as carbon dioxide from the atmosphere. It's going to be sucking up the nitrogen and the phosphorus, however, as ammonia and phosphate ions in the soil that is the substrate for whatever plant. Now, in the ocean, Alfred Redfield, where he was studying, those were dissolved nitrogen and dissolved phosphorus. They can take a number of different forms. I show it as ammonia and phosphate here. Those are the two most prevalent forms, but there's a number of different forms, bioavailable forms that the both organic and inorganic, that the nitrogen and phosphorus can be found in, and we'll discuss that in a little bit more detail in a bit. All right, so and the point with this is that some of the other elements can shift in their relative concentrations in one form of life, one plant life to another. But this Redfield ratio keeps remarkably constant across autotrophs. And so we're going to look at the global cycling, elemental cycling. It's called biogeochemical cycling of each of these three elements in turn here. I think I start with nitrogen. All right, so I was getting ahead of myself there. Let's keep with the history. All right, so nitrogen the time was ripe for the discovery of nitrogen in the late 18th century, and these three guys discovered it all independently. There was Daniel Rutherford, Carl Scheele, and this guy named Cavendish. I've talked about Scheele and Cavendish. We're going to kind of lose us out in the story. So we're going to focus on the way Rutherford discovered nitrogen. He did this experiment with a bell jar. So he took a bell jar, stuck it in a basin of water. Very simple experiment. He stuck the bell jar in a basin of water and just wanted to see what happened. And basically looking at the relationship between the air and combustion, trying to understand what combustion is. Rutherford and the whole host of scientists at the time, like Lavoisier, Rutherford was trying to keep up with the Joneses across the English Channel. Lavoisier was also investigating what combustion was. They were trying to deal with the legacy of Aristotle and the four elements that Aristotle described, which were earth, air, fire, and water. We'll talk about those again in a bit. And the different attempts to prop up Aristotle's notion of the four elements. These notions included something called calor and phlogiston. And both calor and phlogiston were concepts that are fictitious and were debunked by this generation of scientists. Calor was a notion used to explain what fire was as like an actual entity, a physical entity, instead of just being energy the way we understand it now. And phlogiston was some sort of mystical fire-derived component that invested things that were combustible. And when you burned something, something was burnable because it had phlogiston in it. And then when you burned it, it released the phlogiston out into the air. And this is not how we understand combustion now. But trying to really poke some holes in the phlogiston theory, he discovered nitrogen. And our atmosphere is primarily nitrogen and oxygen with some carbon dioxide and a few trace gases chucked in. So he took this candle, stuck it in there, waited till the candle went out, and noticed that the volume of the bell jar changed. The water went up into the bell jar, and the gas that was left behind was incombustible, and in fact, it was primarily nitrogen. This is how there certainly would have been some carbon dioxide in there, but primarily nitrogen. All right, and so with time, he developed a way to fractionally distill nitrogen and isolate nitrogen gas using a mechanism like you see here. So nitrogen itself is, for example, apples give off nitrogen gas, and it retards the oxidation process that happens. The oxidation process is the browning, is the rotting of the maturation and rotting of organic materials in food, so now on modern apple farms, they'll take apples and seal them in large chambers and flood the chamber with nitrogen. However, if you take like a cooler that's not cool, but just a cooler, and you fill it with apples and close it, and then let them sit for a while, as opposed to apples that are sitting in a bin at the same temperature that's open to the air, the ones that are open to the air will rot much faster. And then if you were to measure the partial pressure of nitrogen in the chest with the apples, you would see that the apples themselves had given off some nitrogen gas and had slowed the oxidation process there. As I had indicated on the Redfield slide, everybody needs nitrogen. I said 77, I guess I got 78 as the number on here. 78% of the atmosphere is made up of N2 gas. N2 is this molecule you see over there on the right-hand side. It's a triple bonded nitrogen. I'm putting all these chemical structures on the board. Please don't take this as being condescending. I want to make sure everyone's with me. Are people familiar with basic chemical structure formalism? Like when I draw, you want me to explain that? All right. So let's take the pyrimidine there, that one that's like a hexagon with a couple ends in the middle of it. When I draw this, what we have here, this is what's drawn on the board or on the screen. What I actually mean there is there's a carbon atom here, a carbon atom here, a nitrogen atom here, a carbon atom here, a nitrogen atom here, and a carbon atom here. This carbon, nitrogen are connected by 2 bonds. That means 4 electrons or 2 pairs of electrons. A bond is a pair of electrons. Okay, and there's 1 pair here, 2 pairs here, a single pair, a double pair, a single pair here. Carbon forms 4 bonds around itself. And so when you see this picture, none of the hydrogens are really written in unless you want to explicitly write them in like the case of the amino acid you see there. So in this pyrimidine, there's an implied hydrogen here, an implied hydrogen here, 1 here. This nitrogen has 3 bonds around it, so those are both happy, and there's an implied hydrogen there. So when you see a structure like this is what I'm actually talking about. Okay? Does that clear things up a little bit? If anyone needs more background than that, we can talk later. All right. So nitrogen is found in some extremely important biological molecules in your body. It's a essential component of nucleic acids. And so we have 2 categories of nucleic acids, the purines and pyrimidines. They make up the bases, which are the A, T, G, and C, which are the fundamental code that is DNA. We'll get into that a little bit more later when I talk about DNA in detail. And then they're also part of amino acids. Amino acids, so there you see that amino acid. That's the general structure for an amino acid. Amino acids vary by the R group that's on there. They all have that nitrogen, the way you see it, the carboxylic acid, the C double bond O OH group on the right-hand side, and then the amide group on the left. That central carbon, they call it the C alpha, that carbon bears an R group, and we'll talk about the amino acids in a little bit more detail down the road when we talk about how cells work. But so, for example, let's go. Where did I not have an amino acid on the board here today? Maybe I didn't. Yeah, that one. I was talking about another. Oh, it was. Sorry, I'm getting my too many lectures confused. I was talking about Carl Scheel yesterday to my other class. Okay, so the thing about nitrogen is it's often the limiting nutrient in the production of food. So it's often the element that whatever plant that's growing, its growth rate is limited by the availability of nitrogen. And so because of that, and it's not always the case, but it is typically the case. And I'll return to this idea in a moment. But because of that, it leads to the use of fertilizer. And here is this bag of fertilizer that's strictly nitrogen. This would be something you were using in an extremely nitrogen-limited situation if synthetic fertilizer is your thing. So it can take a number of forms. There are the 3 most common forms. Urea, although urea is kind of a subgrade source of nitrogen. And then ammonium nitrate, also anhydrous ammonia is another source that I didn't put up there. And then ammonium sulfate would be used in soil that needs a little bit of sulfur as well. Ammonium nitrate is like a double whammy nitrogen source because it has both the nitrogen in the ammonia and the nitrogen in the nitrate counteranion. All right. Urea does have a lot of nitrogen, though. It's a nitrogen-dense fertilizer as well, 2 nitrogens. It's just not as bioavailable as some of the other forms. All right. So I talked about the food cycle, and here is the nitrogen cycle and how that lays on top of it. This may make you feel like you're going to freak out a little bit. Don't worry, we're going to walk through it together. The nitrogen cycle is essential to understanding how food grows, the chemistry of food. So let's start at the top with atmospheric nitrogen. This is the N triple bond N, that the N2 gas that is floating around in the atmosphere. It's really hard to take so if I needed nitrogen in my body, I am constantly breathing in nitrogen, right? 78% of the air that I inhale is nitrogen, but my body can't use that N triple bond N. It can't use that nitrogen at all. We have to get it into a form that's bioavailable. And thankfully, there are a number of mechanisms that this can happen by. So when you look at inputs, when we look at ways of, it's called fixing the nitrogen. We're going to fix the nitrogen. Initially, it's unfixed. It's nitrogen gas that's floating around out here. And there's going to be 3 or 4 really ways that we can fix nitrogen, get it into the soil in a form that can be absorbed by plants, by autotrophs that then become a source of nitrogen for the rest of us. Let's see if I have a laser pointer here. I guess I don't. So the first input is via bacteria. So we have 2 types of bacteria that are going to do this. This is on the left-hand side of the screen. There are some nitrogen-fixing bacteria that are symbiotic on the little nodules on various plants. And so you've heard probably that certain kinds of plants enrich the soil with nitrogen. Like, for example, red clover is one. Certain kinds of beans enrich the soil with nitrogen. And it's not actually the plant that's doing that. It's a symbiotic bacteria that likes to grow specifically on the roots of that particular plant and provide the nitrogen for that plant to grow, but then also provide more than the plant needs and condition the soil for the growth of that plant. All right, so there are nitrogen-fixing bacteria that are symbiotic with various legume roots. That legume is bean. It's the word for bean. But then there's also just nitrogen-fixing bacteria that live in the soil on their own. In healthy soil, that is. Healthy soil will have lots of nitrogen-fixing bacteria in there. Those nitrogen fixers primarily fix nitrogen by reducing it to ammonium. And ammonium is down in the bottom there. That's one of the central species of nitrogen that all the different sources converge on. Ammonium itself gets worked on by what are called nitrifying bacteria. And there's a couple different steps there. That's why you see different bacteria. I'm simplifying. This chart is actually remarkably simplified from what it would be if I added all of the chemicals into it. But ammonium then converges on nitrates right there. And the nitrates are the ones that are the most readily absorbed by the bulk of autotrophs out there. What's an autotroph? A plant. That's it. Yep, a plant. So another input is from excrement from various animals. That's an extremely small component of the nitrogen that gets fixed in the soil. But we have this rabbit here who its pellets have nitrogen in them. Has anyone had rabbits before? What do rabbits do? What's it like having a rabbit? Oh, eating a rabbit. No, I'm saying, like, has anyone ever had a pet rabbit? I don't know. No, you didn't eat your pet. Has anyone had a pet rabbit? No? Well, oh yeah, tell me about it. It eats its cabbage. What else does it eat? Say it. It's got 2 P's and 2 O's in it. Poop. Yeah, rabbits eat their own poop. And they do. They do, right? I mean, confirm what I'm saying, right? Yeah, they do. They eat their own poop. And rabbits aren't actually dumb and like so hungry they don't know what else to do with themselves or bored. Their poop, and all of our poop, is really full of nitrogen. You don't absorb all the nitrogen that's in your excrement at all. And so that nitrogen in the rabbit's feces enters the soil and is worked on by the detritivores, the decomposers, the fungi, the other bacteria. They're going to break that down and make that nitrogen that's in the poop in a more readily available form, such as the ammonium. That nitrogen in the feces of the rabbit is in organic form. Maybe it's amino acids. Maybe it's some other kind of nitrogen containing, like a purine or a pyrimidine, or maybe it's whatever. There's all kinds of like amino acids, whatever. There's all kinds of forms that the nitrogen can take in the poo. And the decomposers break it down and send it to ammonium, which then is, again, like I said, cycling up to the nitrates and being assimilated by the autotrophs. So there are 2 other ways that we can get nitrogen in the soil. And the first is a natural method, and the second is an artificial method, a human facilitated method that's inspired by the natural method. The first method, the natural method of fixing nitrogen, is lightning. You may think, wow, well, that can't be that much, but actually a significant amount of the nitrogen in the soil got there through the action of lightning. Wherever there's a lightning strike, lightning oxidizes that. It's such high pressure, high temperature, high intensity reaction that the nitrogen forms. It gets oxidized to a nitrate and is in the soil, and where you have a lightning strike the next year, you could actually expect to have significant primary production from the various plants that may be growing at the strike of the lightning. So then there is artificial fertilizer, and that is fixed with the Haber-Bosch process. And we'll talk about the Haber-Bosch process in a bit, but that leads to both nitrates and ammonium that can be artificially applied to the soil. All right, so here's Fritz Haber and Carl Bosch. Who's listened to the podcast about Fritz Haber? What do you do with Fritz Haber? Good. Well, one person did. 2, you did. You listened to it too. Oh, okay. You've listened to it. Yeah, we'll talk about Fritz Haber on Friday. We'll talk about those podcasts in the next class period. But So I won't tell you too many details about his life, but yeah.
Sorry, I don't want to take you back too far, but
Yeah.
so that process, like with the nitrates and stuff like that, like after the nitrogen is changed into ammonium,
Mm-hmm.
which is most likely usable by
Yeah.
autotrophs, then what happens?
So it goes from ammonium to nitrate. These other what are called nitrifying bacteria essentially oxidize the nitrate, the nitrogen in the ammonium to a nitrate, which is a form that is more readily assimilated by plants. It's a little bit harder for plants to directly take up ammonium, but they will take up the nitrate quite readily. So that's all. It's just if you look at the ammonium and the arrow pointing to the nitrates, you see these bubbles with bacteria there. Yeah, they're nitrifying bacteria. There's a couple species of nitrogen that they go through. There's one called nitrite. You don't need to know about it. I went to some pains to try to edit it out of this infographic. All right, so please listen to the podcast about this guy. I'll give you the short story. Fritz Haber was a Jew living in Germany. And he felt the nationalism of Germany as this like ascendant power on Earth during World War I. They realized that for Germany to actualize all of its ambitions, it was going to need more people. How do you get more people? You get more food, and to feed the more people. But how do you get more food? You gotta grow the food, and you need more nitrogen to grow the food. So we need some kind. This is what he's saying. We need some kind of source of nitrogen, and he realized that the atmosphere is full of it. And in fact, they knew about how lightning strikes the ground and fixes nitrogen. So they came up with this process of essentially creating artificial lightning in this big contraption. I think I have a picture of it in a moment here. I forget the exact. Oh, so 1910 was the day that was the year that they invented the process, and then it went into industrial production in 1913. So you can see here that at that first red arrow, that's at 1913. What's interesting about this, you can look at the blue line is so there's the dark blue line is population, all right, and the medium blue line is N fixation that happens in cropland naturally through the application of whatever, manure, etc. And but at that point, at 1913, the thing that started taking off was the pale blue line. That is industrial N fixation. All right, and that's when they started to make industrial Haber-Bosch derived ammonia. And this is not coincidentally the same point at which, despite a series of global wars that killed off lots of people, this is when the human population blew up. It blew up. We just started pouring nitrogen over the human population, and it to a large degree eradicated. Oh, class is over. Hunger. All right. When I go over like that, if I'm not really paying attention, I guess it's 9:50. It's right on the dot there. I wanted to get you guys to answer a couple questions. We'll do that on Friday. All right, we'll pick up with Haber-Bosch on Friday. Any questions?